So we'll get started. There's 47 slides, which we're probably not going to get to all of them today, but that does indicate that the reaction that we're going to start studying is the aldol reaction, and that has a lot of complicating factors in it that we need to sort of figure out what they are and how we can control them. Before we begin, any questions on the material thus far? So we were talking about forming these bonds from the alpha carbon to the beta carbon. A couple, maybe a diversion here, where the beta carbon in these next two reactions is going to be a heteroatom. So these are classic reactions that everyone sort of learns. The first one is called the haloform reaction, and halo meaning halogen, form being whatever that means. So for example, like CHCl3, that's called chloroform. And it's a classical reaction. In the time before spectrometers, it was a chemical test specifically for methyl ketones. So if you have like an R group, a carbonyl, and then a methyl group, it was a test for that. And the reason it works in that form, the halogen that was used was iodine. And so that forms iodoform, which essentially will crash out of this solution as a yellow solid. So I don't think you guys do that in 256, but it's a common undergraduate experiment. If you're given an unknown, and then all of the sort of test reactions you can run, does it have that functional group in it or not? So with your methyl ketone, and again, this is a special case because we have, and I keep messing these pens up, an alpha carbon and an alpha prime in red and alpha in blue, where alpha is your methyl group. The alpha prime here, as we learned, is non-enolizable. And again, that either means it's an sp2 hybridized carbon or it's a carbon that's sp3 with no hydrogens. So under basic conditions, so potassium hydroxide in water, I2 is added, so that's a solid. You run these in a test tube, you just shake it up, and then over time, this yellow solid iodoform will crash out. And then you could say, okay, at least there's this methyl ketone as part of the molecule. And again, something new that I'm doing this semester on the next slide, I'll show you the mechanism. And I'll highlight in blue. So there's that carbon. That ends up being the carbon in the iodoform. And what's interesting about this is, aside from testing positive for the methyl ketone, that carbon is also the carbonyl carbon is oxidized from the ketone to the acid. And so if you ask the question, where does that second oxygen come from? It's going to come from the potassium hydroxide, and then you have to break an existing carbon-carbon bond, which is pretty unusual because we're interested in forming them, and so breaking them, there's not a lot of reactions we learn where we're breaking carbon-carbon sigma bonds. So how does this happen under basic conditions? And so there's our methyl ketone, potassium hydroxide in water, and then I2. So we form this potassium salt of benzoic acid. So potassium benzoate. If you look at your drink or your food that you're eating, one of the additives is probably sodium benzoate. That's put in there as a preservative. My family eats bunny bread, so if like you go to Walmart, there's all different types of bread, right? So I like bunny bread the best. But you can buy a loaf of bunny bread and it will not grow mold for like 4 months. But if you go to like Loblolly Bakery and you buy fresh bread, like within 3 days, it's gonna start growing mold. So things that are like mass produced, there's always these preservatives in there because it's like, it's gotta go from where it's made to some distribution center to the store, and then, so it's gotta last a while. But at your local bakery, they're not putting all these other things in. That's why it probably tastes a lot better. And so we get H and then the iodoform. So we want to understand how the carbon is ending up in iodoform, how the other oxygen's getting in there, and why does it end up as the salt, K plus, and then benzoate. So we have 3 alpha hydrogens, and since we're under basic conditions, we'll deprotonate to form the enolate. And so the enolate is the nucleophile, and then the iodine is the electrophile. So we reform the carbonyl, the pi bond will attack the iodine. And so we've done a substitution of one H for one iodine. And I'm gonna cheap out and say arrow 2 more times. So each hydrogen undergoes the same substitution. You're forming the enolate, attacking another equivalent of iodine. And so all of those 3 hydrogens have been substituted. And so one of the things to think about in terms of mechanism, this is where we're at, and this is where we need to be. So we have to break that bond. That has to get an oxygen, that has to get a hydrogen. So the only thing that's reasonable is, since we're in the presence of potassium hydroxide, is for that to attack the carbonyl. And so that would be a nucleophilic addition. So what's unique about this mechanism is that negative charge on oxygen will collapse to form the pi bond. And then you really have 3 choices, because since you're forming a bond, you have to break a bond. You could break that bond, but that's going to form a carbanion that's super negative, and using the rules of pKa, we can't do that. We could break the bond we just formed, but then that would lead us back here. Or then we can break that carbon-carbon bond. So that is what ends up happening. And the reason for that is the carbon has 3 iodines, so it can stabilize that negative charge through induction. So normally anytime you see like carbon negative charge, that's like really reactive. But again, since it's I3, it's a little bit more stabilized. And now essentially what's going to happen is an acid-base reaction. We have an acidic hydrogen from the carboxylic acid. So we do a proton transfer. And then end up with O- balanced out by potassium. And then we get the iodoform. So is this used a lot nowadays? Probably not, but mechanistically it's interesting in terms of how you break a carbon-carbon sigma bond, and then the acid-base part at the end. And again, we're sort of on this diversion where we're making a carbon to heteroatom bond, and the next classic one is Hell-Vollard-Zielinski or HVZ reaction. That's going to substitute an alpha H for a bromine. So this uses probably a nasty brew of things: PBr3, bromine, and water. And what is mechanistically different, this occurs under acidic conditions, so it proceeds through an enol, which is neutral. And the reason that this is important is now that you have a good leaving group at the alpha position. And then historically what this was used for was to make alpha amino acids. Because now you can displace, say with a nitrogen source, and then you can ultimately carry that on to an amino acid. And that's how classically people used to make those. Was this, I don't remember, is this Tommy Boy? Zielinski Auto Parts? Not. Maybe that's a little bit old for you all. You've aged out of those movies. Okay, so again, those were sort of two separate reactions as an aside. And now we're going to move into, really one of the most important reactions, the aldol reaction. And so now we're going to start making carbon-carbon bonds. To review my slides a little bit better before we get to that. We actually did, I think one of the last questions on last Wednesday was where we looked at alkylation, to figure out what electrophile to use. So this is one of those, and then we'll get to the aldol. So we know that enolate is the nucleophile, and then we're going to add in an electrophile where we're looking at the beta carbon to determine the oxidation state, that it was an alkylation. So from our starting material, we have this issue of alpha prime not equal to alpha. We found that with sodium hydride, that forms thermodynamic enolate, so that's more substituted. So that's specifically going to go after alpha prime. And then we're treating with an electrophile where that carbon bearing the leaving group is at oxidation state 1. And so we're forming a C alpha prime to C beta prime bond. So I'll highlight that in red. And then notice that carbon bearing the leaving group gets reduced. So it goes down 1 oxidation state. And since it's at 0, that's how we know it happened through alkylation. Just to highlight, the mechanism over all of that is SN2. So you want to make sure that your electrophile is following that trend of methyl primary or secondary. You can't use a tertiary alkyl halide because it won't react under those conditions. Have you ever heard of SAM-E? SAM-E. So anyone work at CVS or Walgreens or anything? Freedom Pharmacy, anything like that? If you go to like the vitamin aisle, you'll see that. And so this is like a holistic. That's used for depression and anxiety and things like that. But SAM-E is what nature uses as a methylating agent. So if nature wants to put a CH3 group into a molecule, it's going to use that. So in this class, you're used to seeing like CH3 iodine, like where iodine's the leaving group. In S-adenosylmethionine, you're like breaking that bond, and the whole rest of that molecule is the leaving group. So, nature doesn't use methyl iodide like in organisms because it's pretty toxic. It will alkylate pretty much anything. And again, if you go further on in your studies, things have to be soluble under biological conditions. So the structures of these things you'll learn more about in those classes. If we have time this semester, we'll talk about this base, this sugar, in sort of, just call it a linker. The features of those things and why they're in there to get like the analogous reactivity. And then you see the leaving group is shown as this homocysteine molecule to the right. I have more stuff in the middle here. So one more thing that's important to talk about is vinylogous acidity. And I don't want to say I completely lied to you by saying only the alpha carbon was acidic. There's something called the principle of vinology. Any Pearl Jam fans in the audience? Okay, so you won't get that reference either, but vinology is when you take any functional group and then you stick a double bond in there. It makes it vinylogous. So in this molecule shown, you can see a ketone. So you have the alpha prime and beta prime, which are a double bond. So overall, we would say, since that's an alkene plus a ketone, that the molecule is an enone. And then furthermore, you would just shove in front of that alpha prime, beta prime enone. The regular alpha carbon that I've circled, that's the normal sort of acidity that we've seen. If we're going clockwise, so alpha and then beta, we said that beta was not acidic, right? But if we're going the other way, so counterclockwise, alpha prime, beta prime, and now that's gamma prime. Just depending on which way you're going using that nomenclature. That carbon actually does end up being acidic because of that double bond. And it says, why is that? So let's just say we had a base that deprotonated that. So it would form that. But we know that when we have a lone pair or a radical or a carbocation next to an alkene, that you can form a resonance hybrid. So if you do that, so that now looks more like you've deprotonated the alpha carbon originally. So that resonance form is what you're used to seeing as the enolate. So again, depending on which way you're lettering things, the gamma carbon can be acidic if there's that double bond between it and the carbonyl. And I'm not just showing you this as a one-off. Later on in this module, we're going to look at like that specific collection of elements, a carbonyl and a double bond. That has its own unique reactivity pattern, which will introduce like 3 new reaction conditions to us. I feel like I'm saying, but so here's something I like to think about. So if we were going to treat this with NaOD in D2O, how many total hydrogens would be exchanged for deuterium? So we know that these regular alpha ones, those will exchange right away. And then we just said that the these gamma ones will also exchange due to that vinylogous acidity. But also from that resonance form where that looked like an enolate, that H will exchange too. The only one that doesn't get any love is that beta prime one. So this one remains an H. There's no form at which the negative charge would reside on that carbon. And so if you see something like that on your next exam, really what's changing is maybe, is it a ring or not a ring? What's sort of sprouted off at these different carbons? And you're looking for how many deuteriums you can get into the molecule, and then what would the molecular weight be? Leading up to So without further ado, we will start the aldol reaction. And I want to give you just a bit of context about why this has been studied so much. And so very briefly, what I'll lead you through is marine natural products. So again, a natural product is just something that an organism is producing. And typically they're called secondary metabolites. As you'll see in the example today, these are pretty complex molecules. And so if you read the older literature, the titles would always be the chemical constituents of so-and-so plant or sponge or whatever. And they end up being pretty complex. And part of that complexity is they can be used as either drugs or starting points for drugs. And a lot of these end up being pretty cytotoxic, so anti-cancer agents. So, this sort of explanation is focusing on marine environments, but people do this on land as well. Probably the marine environment is more interesting because they go out and scuba dive. Any scuba divers in the crowd? Anyone? No one? So really what they're looking for are species that haven't been studied as well, and they'll harvest these sponges from the sea, wherever it may be, and then they'll freeze dry it. So that's called lyophilization. It's just a technique to get the water out, and then they extract it with organic solvent. So again, the majority of students are biological sciences majors, and if you're not aware of, there's these really well-known oceanographic institutes. They don't just deal with ocean sciences. I mean, these are like huge research enterprises. So Cold Spring Harbor is in Cold Spring, New York, on Long Island. Scripps is in La Jolla, or if you're unfamiliar, La Jolla, California, or Florida. And then Woods Hole, I think, is somewhere on the East Coast, Virginia or something maybe. And I guess there's another one, Harbor Branch. So there's 4. And the reason I bring that up as well is, most of you are, sophomores or juniors or whatever. If you're looking to do research, You can go on their websites, and typically they'll have summer programs that you can apply for, like 10-week programs. You get paid. It's $5,000 to $8,000 for the summer. Normally they take care of your travel and all that stuff, but it's a good way to get out of the SIP, earn some dough, and then get some research experience. And so I can't click on that because this is just a PDF, but if you go to that YouTube video by Paul Jensen, I think you'll find it pretty fascinating. It's, a 25-minute talk or something like that, but he talks about, going into the ocean and getting these organisms, getting the molecules out, and then testing them against various cell lines and cancers and stuff. And that in itself is, it's pretty fascinating. So it's from sea to pharmacy, if you just stick that in your YouTube search. And so the molecule we'll talk about in relation to this chapter is called discodermalide, which, back in the 90s and early 2000s was sort of the hot topic in organic synthesis. Not only due to its complexity, but it's a potent immunosuppressive. And like I said, a lot of these things end up being anti-cancer agents. And what I mean by potent, qualitatively, if you go on to take like medchem or pharmacology or toxicology, this is like nanomolar scale. So really a dose of this stuff is like a milligram, like 1 milligram. Your average pill at a pharmacy might be 5 milligrams, or if it's an antibiotic, 500 milligrams. So you don't need a lot of this stuff to treat a lot of people because it's so potent. And so it was isolated from this marine organism. I'm not a biologist, so I'm not going to try to say that name. But again, they get on a boat, scuba dive, look for stuff like, hey, that doesn't look familiar. Let's grab some and study it. And so this was off of Luque, Grand Bahamas. Can you imagine? You're doing science in the Bahamas with your Mai Tai on the boat. It's interesting, if you read the papers, they'll talk about the depth at which they find these things and all of that stuff. You don't have to be terribly versed in, I need to take Bio 102 before I can read this. It's written so that you can understand it. And so if you look at it, it's a pretty big molecule, and it might look complex. There's a lot of functional groups, oxidation states, stereochemistry. But as we learn this reaction, you'll see that it's actually not that complex in terms of the bonds you have to make. So this is a slide I show to not intimidate, but to really make you aware of the complexity of how nature does this and how everything is sort of intertwined. And so this is a metabolic metro map. And, these are all these cellular cycles, but really what they are organic reactions. The bond making and breaking that you've learned in this class, it's represented on this slide, but it's carried out by enzymes. And so as it says, just a brief detour, how does nature start doing these things? And so nature works in very small units in a sense, but the molecules are large. So this is acetyl coenzyme A, or acetyl CoA. And what is the gist of this? Remember in the prior module we were learning about, these oxidation state 3 functional groups. and we were substituting X. So this is what nature uses. The acetyl are these two carbons here. So nature will start with a 2 or 3 carbon unit, and you see the sulfur and everything else bonded to the sulfur. That's the leaving group. And we see the alpha carbon. So you can do things at the alpha carbon, then you can do substitutions where the sulfur is. And again, that pantothenic acid is a linker to the diphosphate, which is bonded to the sugar, and then that nucleoside base. So that's 2 carbons, or nature will use propionyl coenzyme A that has 3 carbons. And then the coenzyme A is just the rest of the molecule or the leaving group. So if you were in like my research lab, you would never use like a thioester because they're like really reactive. So normally we just use esters, but nature uses these because of that lower bond association energy. So this bond here is easier to cleave. Plus with the coenzyme A, that's a huge stable leaving group. You would never want to use that in lab because your NMRs would be a nightmare with that big part of the molecule, and you're only doing something really on the left-hand side here. So the secondary metabolite that you saw, discodermalide, it's created through a series of enzymatic reactions. And since there's a lot of alcohols, this is called polyketide. So polyketide synthetases. And as you learn about cellular and molecular biology and genetics and those disciplines, how can like those 2 carbons of that acetyl coenzyme A become discodermalide? So the PKSs have modules, which are collections of domains. And in the domains, individual reactions occur. So just think of like what we've learned, but in a cell setting. So the modules either start or load, elongate or extend the chain, and then terminate or release. So I'm going to have a very crappy drawing here. So the ball is like the anchor in which that carbon chain is bound. And then you get elongation of that chain. And really you're undergoing additions through oxidation or reduction. And that's how you grow the chain out. Then it gets to a certain point, and then it's released from that ball. And so to explicitly show that, this is from the Roche website. So they're a large biopharmaceutical corporation. And again, if you go to that website and look at this, you can pinch and zoom on this thing so you can if you looked at this whole thing, you couldn't even read it. It's so big. So you have to like pinch in pretty far to see what those cycles from that like metabolic metro path on the other slide were about. But in here, it shows you, the synthesis of coenzyme A. And then, how that's going to then enter or leave different metabolic cycles. But if you are moving on, it's just organic chemistry. The molecules just get bigger, but the bond constructions you've learned are the same. So I want to start talking about these oxidation state relationships and the stereochemical relationships in discodermalide. So the first thing I want to do, see this lactone? So that cyclic ester. We're going to break that bond and then just unravel the chain so it's in the normal zigzag fashion. So maybe I'll color it in. So when we do that, that blue oxygen is that one right there. And again, that's a module 3 reaction. You took an ester and you saponified it to a carboxylic acid. So that's a common thing organic chemists will do is let's break any rings we can. Let's make things acyclic. So now that it's acyclic, in the red box I've highlighted 6 carbon atoms. And the question is asking you, where do you think those come from, the acetyl or the propionyl coenzyme A? So remember, acetyl provides 2 carbons, propionyl provides 3. So this comes from propionyl. So if you look 1, 2, 3, 1, 2, 3. And we as humans know this because biochemists have used radioisotopes to label specific carbons. So with like carbon-14, if you know where you're putting it, you can then trace it along the metabolic pathway. So I've highlighted that carbon, I'm going to call it 1, and then just counting over to where the red arrow is, that would be carbon 3. So we're going to use a lot of different numbering. It's just we need to understand in what context we're numbering those things. Like what I'm talking about right now is the relative relationship of those oxidation states. So carbon 1 is an acid. Carbon 3 is a secondary alcohol. So I would say overall, they're in a 1-3 relationship. if we kept numbering 4, 5, 6, 7, what you'll notice is that only the odd numbered carbons have a hydroxyl group, right? 1, 3, 1, 5, 1, 7. So in the way that this stuff works in nature is that the 1n, where n equals an odd number, that's called a natural bond disconnection. And then if the number is even, like 1, 2, 1, 4, that's unnatural. And what that gets back to is how you match the polarity of the functional groups. And that's really the underlying thinking of the aldol reaction. So many people over the years have developed their own teaching philosophy of this. I prefer Professor David Evans. He was at Harvard. He passed away a couple years ago. But he called this charge affinity analysis. And what that boils down to is what we learned in Organic 1 about electronegativity and assigning delta plus, delta minus to the bonds. And his famous saying to his students was, go to the board, to spark discussion of things. So it wasn't necessarily, hey, I want to prove you wrong or anything, but let's talk about what are the possibilities from this analysis. And an aside, this website is called My Chemistry. And so it's basically a genealogy. And so Evans is here. My PhD advisor was David Hart, and I'm right there. And these are my 3 PhD students. So I think I was number 40 from Hart's group, and Evans probably has from that door to this wall in terms of the number of PhD students that left his lab. But it's interesting to think about how knowledge gets passed from one generation to the next. Evans, I think, again, was very influential in a transformation that happened in organic chemistry due to his study of the aldol reaction. So he started off, I think, at UCLA, moved to Caltech, published his aldol work, and then moved to Harvard. So what is it in practice? Let's look at propanoic acid. And I've put these, normally we write those as formal charges, but we're just going to call them the charge affinity for each oxygen, carbon, and the one hydrogen in there. And again, that's just based on electronegativity. When you do that analysis, it helps you understand what bonds you can normally make. And then through that, how can I then disconnect things to bring them back together to make the natural product or whatever molecule. So in the molecule, if I can point out, see this negative charge right here? So we know that's an alpha carbon, right? And we know how to generate an enolate. And then you see its match right there, which is a beta carbon. So if we want to make that alpha-beta bond, we have to use an electrophile to attack that beta carbon. But see how it's at an oxidation state of 1? in our starting material, we're going to bump it up to oxidation state 2, so that when we do form the bond, it gets reduced to 1. And the other thing that's bonded to there is an H. So if we think backwards and oxidize it, we would end up with an aldehyde. So the aldol reaction is an enolate attacking an aldehyde to make the alpha-beta bond. And then once you understand that, the whole rest of this molecule is just an iterative process where you repeat that over and over again. But you run into this, right? You have an alkene. You can say, well, there's no alcohol there. But an alkene really can be made from a dehydration of H and OH, right? So you're just dehydrating an alcohol. That's how you get the alkenes. So to show you the effort of what like university groups do. So this structure was published in 1990, and then a couple years later, people started to publish synthetic routes to it. And I can say that synthetic organic chemistry is just as competitive as college football. I mean, you can see all the different names on here, right? These people put a lot of effort into trying to make this stuff, not only because they want to plant the flag on top of the mountain, but again, since it is so potent, if you develop efficient route to it, I mean, you can help a lot of people that have, breast cancer or pancreatic cancer or whatever. But the thing to note is look at the total number of steps and then the overall percent yield. So this is not your 3-hour Tuesday night in Organic 2 lab. I mean, these are like really long sequences. So if you had to start from something and carry that material through 20 steps, if you started from a gram and every step gave you conservatively 50% yield, so a gram, 500 milligrams, 250 milligrams, 125 milligrams. By the time you get to the 20th step, you don't even have a balance that can weigh out, those couple molecules you have left. So the scale in which you start, you're starting with 100 grams, 500 grams, because you're taking these hits along the way. So, as I said, we'll definitely not get to 47 slides. But we'll introduce all the complicating factors of the aldol reaction and why we want to control it. So the first case we're going to look at is with ethanal. So the 2-carbon aldehyde. And then we're going to say this is the self-aldol. That just means 2 molecules of this will react. And we'll use basic conditions. So sodium hydroxide and water. I think I have a blank. Okay, so what we're going to do on the next slide, from those 2 molecules, we have to form an enolate. And the enolate is going to react with the electrophile, which is just the aldehyde itself. And so we'll push the arrows for that, and we'll look at what is formed and what the general form of that product is. What are you looking for to determine if it's an aldol reaction? Slide. So I'll say two equivalents of that, but it's just like, okay, you have that in there in excess, you're adding base. So under basic conditions, we'll form an enolate. So here's the resonance form. So again, that's the nucleophile. I'm going to try some magic here since I need some room. I think I want to do that. All right, I don't know how to use this app, but I'm going to redraw that just because I need some more space to show some things. So again, that's the enolate. There's your alpha carbon. Now a second molecule of that ethanol is going to approach it. And so this is going to be your new beta carbon. So that will collapse to form the carbonyl. This will attack that aldehyde carbon. We break that pi bond. So we made the bond between C alpha and C beta. Notice, I'll use in blue here, that started at oxidation state 2. It's now going down to 1. And the general form for an aldol, so beta hydroxy, I'm just going to say carbonyl. So the carbonyl can be an aldehyde, a ketone, an ester. So again, that carbonyl can be whatever, but the beta position has to be hydroxy. And then to finish it off is the proton transfer. So this would be 3-hydroxy butanal. So just as a review, the parent name comes from the highest oxidized carbon, which is the aldehyde. So that's why it's named as an aldehyde. That carbon's at a lower oxidation state, so it's named as a substituent. So what is one of the complicating factors in here? So carbon 3 is a stereocenter. So really what you've produced is a 1-to-1 ratio of the R and the S enantiomers. And so since it's 1-to-1, it's a racemate. If you put it in a polarimeter, the specific rotation is going to be 0. Can't remember the number of stereocenters in discodermalide, but you know, if it was let's just go back and count them. So 13. So what is 2 to the 13? All right, where's the button? So I think my calculator, I did it right. If I did that right. It's saying 8,192 possible stereoisomers. But when that molecule is isolated from the plant, it's only that one. Like, that's the absolute structure. So if you can, make the mental leap from this to that, if you're in the lab, how do you control that? You don't want a racemic mixture. You want to produce R or S. And then it grows exponentially as you start adding more stuff to the starting materials. So just to recap that over the next couple slides before we get into the next complicating factor. With ethanol, your electrophile is the 2-carbon unit, so that's going to eliminate A and D. Your enolate, again, since it's ATE, must be negatively charged, so that will eliminate C because that's an enol. So B would be your correct answer. So the molecule, the beta carbon, is a stereocenter. So we have 2 to the 1, we get 2 stereoisomers. And as I said, they have to be formed in a 1-to-1 mixture because there's nothing else that's going to discriminate or control that distribution. So the aldol product is the beta hydroxycarbonyl. And as I said, when you see that double bond in the chain, what can happen is a dehydration from the alpha to the beta carbon. So you're losing the elements of water. So H and OH to form that double bond. So it says typically the E-alkene geometry is preferred, so that's the trans. From a simplistic point of view, the trans is obviously more stable because of less steric interactions. But I do believe we have a slide to draw that out. And depending on how good I can visualize this without a model, may or may not get it right. So other than just saying, well, we're going to get trans, why is that? And so we're going to bring back our friend, the Newman projection, because we have to eliminate the H and then the OH is going to leave. So we need those to be in what, remember the word, the orientation of them? They need to be anti. And maybe what we'll do is we'll put our eyeball there and we'll look down carbons 2 to 3. So I want the aldehyde group up. No, sorry. I knew I'd mess this up. I want the H down because it's pointing down already. And then we're going to sprout off those other two groups. So CHO and then H and then the back carbon. If we have OH up, methyl and H. So that would be the preferred Newman projection conformer to do that elimination, again, because the H in the OH are 180 degrees or anti. And then when you do that elimination, I'm just going to flatten these things out. That would be like a Newman projection looking down an alkene bond. And then if you rotated your eyeball, you would see that those groups, the methyl and the aldehyde, are trans to one another. So maybe just a couple more minutes and then we'll end at this point to show you why that's preferred. So I was plucking off this H right there, but what about this H? We would have to do a bond rotation to get that H anti. So we'll indicate that bond rotation. I'm going to leave the back carbon the same. I'm going to rotate the front one counterclockwise. And so you see how the colors of those hydrogens moved. So now the blue one is anti. So if you do that elimination, man, these stupid colors. They changed this app, so I'm dealing with how things move and look different. So we have OHC, boom, boom, H, CH3. And so now you can see those two groups are cis. Why that would not be preferred again is you have this steric interaction here, whereas over here it's minimized. And so in general, that's why you would prefer the trans geometrical isomer under those conditions. So I think we'll wrap up here for today, because the next thing, it's going to get more complicated when we're not doing a self-aldol. If you look, we have two different carbonyl things in there. And so then now the number of products that we can produce is going to be a lot more. And so we'll look at that. And then on Wednesday, we're going to look at, okay, we don't want to produce a lot of things. How do we control these factors to get into one stereoisomer? And there's ways we can do that, again, with the base that we use, what type of carbonyl we're starting with. And then I'll give you a little bit of history of how Evans started to control that, and then how one of his students took it to the next level and actually won a Nobel Prize for it. So that'll be on Wednesday.